Let's take a look at how we design PCR primers. In the first exercise, we will design primers to amplify the segment in blue. You're looking here at double-stranded DNA. The two ends of each strand are not chemically identical. You have a 5' end and a 3' end. First of all, let's separate the two strands and imagine the direction the DNA polymerase will be moving in to synthesize the new strand by complementation. You may recall that DNA polymerase always reads the template in the 3' to 5' direction and adds nucleotides by complementation in the 5' to 3' direction. So let's take a look at the top strand first. In this case, the DNA polymerase will be moving from right to left. And to copy the piece of DNA in blue, it will first read out G and add C by complementation. Then it will read out A and add T. It will read out A and add T, and so on. And therefore, it is now easy to imagine that the primer that DNA polymerase will need to start amplifying this region will have the following sequence. At the 5' end, it will have C, T, T, C, and so on. And for sequencing, we typically use primers between 18 and 24 nucleotides long. Now let's take a look at the bottom strand. In this case, the DNA polymerase will be moving from left to right. And in order to copy the piece of DNA in blue, it will start with C and add G by complementation. Then it will read out C and add another G. Then it will read out G and add C, and so on. And therefore now it is easy to understand that the primer that you would need for DNA polymerase to be able to copy from the bottom strand would look something like this. And for DNA sequencing purposes, again, we would choose a primer anywhere between 18 and 24 nucleotides in length. In this scheme of things, the primer that goes from left to right is called forward primer. The primer that goes from right to left is called a reverse primer. By convention, we write down the sequence of the primers always from 5' to 3' direction. So the sequence of the forward primer written down in the 5' to 3' direction will be exactly as you see in this figure. Now the reverse primer is a little bit more tricky. Here, if you read the letters from left to right, you will be reading from 3' end to 5' end. And so to show the sequence of the reverse primer in the 5' end to 3' end direction, we actually have to read from right to left, and it will read C, T, T, C, 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 and so on. Now, the convention is actually to display a nucleotide sequence of only one DNA strand, with the 5' end on the left and the 3' end on the right. So let's remove the bottom strand and display the DNA sequence as we normally see it in publications or exercises. And let's take a look at how the sequences of the forward primer and reverse primer now relate to the sequence of the DNA fragment that we are amplifying. And now you see very clearly that the sequence of the forward primer is simply the first 20 or so nucleotides of the DNA sequence that we are amplifying, and the sequence of the reverse primer is simply the reverse complement of the last 20 or so nucleotides of this DNA sequence. It is as simple as that. So what makes a good sequencing primer? There are three basic rules that you need to remember. Sequencing primers are typically between 18 and 24 nucleotides in length. The 3' end of the primer should be either G or C. This creates the so-called GC clump, and we do so because the GC bond is stronger than the AT bond. This ensures strong binding of the primer to the DNA template at the 3' end of the primer. And finally, the melting temperatures of the two primers, of the forward and reverse primer, should be within 5 degrees Celsius from each other. So now that we have gone over the rules and considered one example. Let's design PCR primers to amplify this nucleotide sequence. So to draft the forward primer, all we do is we start with the first nucleotide and take the first 20 or so nucleotides, and we end these 20 or so nucleotides with G or C to ensure that we have this GC clown. So our draft sequence for the forward primer will be simply the sequence shown here in red. Now, to draft the reverse primer, we start with the last nucleotide, and counting in reverse, we take the last 20 or so nucleotides, and again, we aim to end that with G or C. Then we copy-paste this sequence into any server that generates a reverse complement. For example, you can use this bioinformatics server. So a reverse complement of the last 20 nucleotides will be this sequence, CCG, AAG, and so on. So this is our candidate for a reverse primer. And now the final step is for us to compare the melting temperatures of the forward and reverse primers and adjust their length if they differ by more than 5 degrees Celsius. In this case, I used Thermo Fisher Scientific calculator of melting temperatures of primers, but if you simply Google calculate melting temperature, you will find these kind of calculators available from other major pharma companies, such as, for example, Promega or NEB. Any of them is good. So in this case, we have been lucky. You can see that the melting temperature of the forward and reverse primers differ by only 3 degrees. This is fine, so these primers are good to be used in the PCR. If there was a difference in temperature of more than 5 degrees, then we would try extending or shortening either the forward or the reverse primer, but we would do it always at the 3' end. If we extend or shorten the candidates for forward and reverse primer, we do that at the 3' end. So I hope this has been helpful.